Thank you very much for inviting me. As you've heard, I'm a chemist. I'm not a dyer, and so my approach is a little bit different. And so my interest was trying to figure out what people used for dyeing in ancient times, and the ancient means more than about 500 years ago. Going back, well, the earliest thing we've seen is about 3,000 years old. Let's see if I can figure out how this works. This one. Now, unfortunately, if you want to look at really old dyes, there are only a few places in the world where you can get samples. And they tend to be places that are pretty dry. And that includes, I still have to figure out, okay. This area going from Japan to all the Middle East, and also in Peru or in the Andes Mountains, which is also very dry. The problem is that dyes are, and textiles too, are organic compounds, and they tend to be decomposed by microorganisms. And all microorganisms need water, and in parts of the world that are humid, there's just not much left. So what you need here are places that are dry, and one way to get dryness, of course, is to live near a desert or someplace that's naturally dry, but also many tombs, well, some tombs anyway, stay dry because they eliminate water. Now, looking for dyes or textiles is sort of the same as if you're storing meat. You want to keep it dry or cold. There are a few cases where materials have been frozen in ice, or you can have lack of oxygen because microorganisms need oxygen. And so some peat bogs in northern Europe have low oxygen, and so there's some examples there, although I personally have not analyzed those. And then we also have use of preservatives, for example, salt mines, because you can store meat by salting it. Now, I just wanted to talk about sort of two approaches to analyzing textiles. One is you shine a light of some sort on a sample and then analyze the reflected light, which is over here on the left. And the other approach is to actually take a sample and extract the dye from it and then analyze it. And this is the technique that we use. And comparing them, you can get much more information by this approach. You can see that you get, and the important thing is separation of the components. Here, if you shine light on something, the result you get is a mixture of all the components on the textile. The downside is, though, you have to take a sample. So you have to cut out a piece and analyze it. another important factor is remote analysis. If you're using this approach, you either have to bring your instrument to the textile, or you have to bring the whole textile to the instrument. And sometimes this is difficult. With the method that we use, and most people use actually, you can just take a small sample of the textile and send it off and have it analyzed. And the approach that or the methodology we use, which is becoming fairly standard now, is to take the dye extract, run it through an HPLC column to separate the components, then analyze it with a diode array detector and a mass detector. Now, if you don't use the mass detector, you use only the diode array detector. Well, first of all, you get a profile that looks like this for any extract. This happens to be from a pagoda tree bud extract. And then you can get a spectrum of each component here. And that tells you usually what type of dye it is and what category of dye, but it doesn't necessarily tell you very much more than that. If you use the mass detector, then you can get the molecular mass of it, and that eliminates many possibilities. For example, flavonoids mostly have this type of spectrum, but there are 8,000 known flavonoids. There are probably many more out there that have never been analyzed, and they all look sort of the same. So, but by using a mass detector, you can eliminate many possibilities. Now, this particular case, this peak here is rutin, which is a derivative of the flavonoid quercetin with a couple of sugars attached. Now, we've chosen to focus on yellow dyes because there are lots of them, and they grow all over the world, and so dyers can often find a yellow dye nearby. And so that, if you know where that plant grows, then you can make some guesses about where the textile is dyed. And there's a few red ones, but there are really not so many stable red dyes. There's light, fast dyes. A couple of purples. We'll hear about one type a little later. And there's only one blue. It's indigo, which is wonderful for dyers, but it's not so useful for what we're doing. And browns and blacks, there are just simply no good ways of analyzing those. So it would be interesting to do, but we can't do it. So yellow dyes first. Now this just shows some typical yellow dye plants in different parts of the world where you see them frequently. Weld is, at least in the last 500 years or so, has been very widely used in Europe. But in Central Asia, we found this yellow larkspur, which is Delphinium semibarbatum, and it has a very distinctive profile, and you don't see it anywhere else. So it's actually fairly distinctive to tell you that it came from Central Asia. In Xinjiang, well, I talked a little bit more about that, but one possibility is poplar leaves, which poplar trees grow there in quite abundance. And then, well, the pagoda tree was used in China a lot. And then in China and Japan, at least in ancient times, they used some grasses, arthraxin, and these are flavone dyes. And finally in Peru, we don't know so much about that. There hasn't been much published. But what I want to point out here is most of these yellow dyes are flavones or flavonols. And the flavone from weld was used in Europe a lot, but in other parts of the world we find a lot of these flavonols. And this just distinguishes between this overall structure of these compounds, flavonoids, is the same, except for this substituent here on position 3. In weld, it's a hydrogen, and this is fairly light fast as yellow dyes go. But if you put an OH group there, it rapidly decomposes in the presence of light. However, if you put a sugar on that, substitute the hydrogen with the sugar, you get something that's fairly stable to light. Now, what I want to do now is just give a few examples. Many of them we've looked at, but I'll just give a couple of examples. One of the first things we actually looked at was some samples from Xinjiang, Chechen, which is the cemetery where sort of a famous mummy was found. This guy is 3,000 years old, and they just put him a hole in the ground, but because it's so dry there, he was mummified with no special treatment. He actually looks in pretty good shape for somebody who's 3,000 years old. But buried with him were some textiles, which are shown here. And we got some samples. Irene Good, who has participated on this study of these things in the 1990s, was able to get a few samples out of China, very small samples. But one of the ones we found, we looked at these yellow dyes especially, but we found something here that was different from what we'd seen before. Now, if you look at this slide, this is there's a big luteal glucoside peak over here, which I didn't put in, but if you look at the smaller peaks, you get this pattern for weld. This particular yellow here may be weld, but this one definitely is not, because this big peak 4 comes over here. So there's something different there. And it took us actually several years to figure out what it probably is. It has the glucuronic acid on this position with the carboxyl group here. And we've only seen that in samples from Xinjiang. Excuse me, I'm getting kind of dry here. Now, another sample we got is from Iran. There's a salt mine there that's been worked for at least 1,500 years. they found several mummies there, 5 or 6 of them now. And apparently what happened was that these poor people were working in the mine, and there was an earthquake occasionally, and everything came down on top of them. And the good thing for us is they're well preserved because they were stored in salt. It's also fairly dry. So this is the same as this here. The ceiling just came down on top of it. Well, we got a sample, not from that particular mummy, but from sort of a belt that was found. And I asked if we could get a sample for analysis. And the archaeologist, who was not a conservator, gave us a sample. This is the sample here. You can see where it came from. It just took a big pair of scissors and cut it out. Well, it turns out that this particular yellow is something that we'd never seen before. I don't think anybody's seen it. In that it contains, it's a flavanol, but it has a sulfate on this position. This is the salt mine here. And we've analyzed a lot of different samples of tamarisk from around the world, and they all have these flavonoid sulfates, which are fairly unusual. And I won't say that this yellow is commonly used in Iran, but it's sort of unique. And I mentioned earlier that in Central Asia, the yellow larkspur is used a lot. Usually it's a delphinium. Usually you think of delphinium as being blue or purple. This is bright yellow. But these are all flavanol 3-O-glucosides. And then we have the protoberberin type dyes from the cork tree. These are not flavonoids, they're very different. And the dye comes from the inner bark of the cork tree. Here you can see it being peeled off. And these things are used in medicine in China and Japan for centuries, and they're also used for dyeing. Now, usually people talk about, when you see a protoberberin, say it was dyed with the amber cork tree, which may be true, but there's also another species of philodendron that grows in central China. And so this might have been used too. The problem here is it's not always easy to distinguish between them, because even with, say, the cork, with the Amberenza, it depends on which part of the tree you analyze. You get different patterns. Or sometimes the south side is different from the north side of the trunk. So it's a bit of a problem. And I'll just say a little bit about red dyes. The major red dyes that are light fast are madder. Well, they're madder. Safflower gives a beautiful red color, but it fades pretty easily. But it's possible to distinguish at least 3 of these species of madder. And I'm just going to talk about the plant. These are plant dyes. We also have insect reds. And we've sort of concluded, at least from our studies, that these 3 species, Rubia tinctorum, is universally found in the sort of the western part of Eurasia. Rubia cordifolia is sort of famous for being called Mungistan, is from India, and I don't know exactly how wide this range is. But, and then the third one is Rubia acana, which is, at least nowadays, mostly used in Japan, but you also see it in here sometimes too. There's some subspecies or minor species of matter that also have the characteristics of Rubia acana. And by HPLC, it's pretty easy to distinguish them. Rubia tinctorum is the only one that has a lot of alizarin. They all have purpurin. And Rubia acana has an entirely different anthraquinone color, and these are glycosides. But these three groups can be distinguished. The problem here is that Rubia peregrina, which is found around the Mediterranean Sea, and Relbunium, which is found in Peru, they're pretty much the same as this. They have just the purpurin color. Now, I showed that map of the three species of matter. We got a sample from, well, it was found at Laursen, which is in Xinjiang, and analyzed this purple. This purple is made from indigo plus, turns out to be lac, which is an insect red. And so it came from here. And lac doesn't really grow in that part of the world, so it must have been brought in from someplace else. And we don't know for sure, but our guess is, because lac insects grow down in more tropical regions, it was probably imported along here, because the old, this is the Silk Road that goes this way, this one version of it, but there was also a branch that went down south to India, so it may have been brought up through here. And then finally, South American dyes. They have some beautiful colors. There's a mummy, it's probably 1,000 years old. Bright yellows and reds. And we didn't actually analyze this one, but we did analyze a few samples from these two places. I'm not going to go into what we found there, but except that there's been very little study actually of these dyes from this part of the world. The samples that we did analyze looked like flavonoids, and we found some chalcones, which we've never seen in anywhere else. I'm not sure why that is. And then the red, commonly used Relbunium, which is a type of matter, or it's gallium actually. It's matter-like, and cochineal, which they probably got from Central America someplace. So this is just to summarize what we found. We think we can get clues to the origin of a textile, and where there's no documentation, but it's only one clue. You have to use other information. And as I said at the beginning, our sampling is limited by where you can get samples, and that tends to be in arid areas. Maybe it's just my personal feeling about this, but I think you can get more information from yellow dyes about location than you can from some of the others. So that's really all I want to say, except to acknowledge the people who worked on this. First is my last graduate student, Shen Zhang, who actually started this project, and Chika Mori, who was with me for a couple of years, and our organizer here, Jen, spent a few months in Boston, and brought a bunch of his samples, which maybe you'll talk about them later. So I think that's it.